This is a special episode of the Stem Cell Podcast, ISSCR 2024 on day one. Hey, everybody. We are Doctors Daylon James and Arun Sharma. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field, literally in person here at the meeting. Yesterday launched the start of the 2024 ISSCR annual meeting in Hamburg, Germany, and Arun and I and the whole Stem Cell Podcast team are attending the meeting. Drop by the Stem Cell Podcast booth in the exhibit hall for a chance to win some prizes and to find out how you could be featured on the podcast. You can also meet Arun and me at the Meetup Hub in the exhibit hall. That's on Friday, July 12th at 3:45 p.m. local time. We'll be hanging out and chatting with the listeners. And Friday is going to be a busy day with our very first live show happening at 6 p.m. local. We can't wait for this live recorded conversation with members from the International Space Station National Laboratory. Today and every day throughout the meeting, we'll be releasing special episodes discussing our favorite sessions and events at ISSCR 2024. So if you weren't able to attend the meeting, we've got you covered. We're going to kick things off in just a minute, but before we get to that.
Are you currently attending the 2024 ISSCR annual meeting? STEMCELL Technologies wants to give you the chance to win one of 3 awards of $500 to offset your travel costs. Enter at www.stemcell.com/forms/ISSCR-travel-award by Friday, July 12th for a chance to win.
With all that said, let's go, Arun. Here we go. Kicking off ISSCR 2024, which has been a long time coming, to Hamburg, which is also known as Germany's second city. That's a fun fact. The meeting actually started with a few words from Hamburg's second mayor, Katharina Fegebank. I don't know what a second mayor actually. It's like a vice mayor, but she had some really inspiring words to share about the cross section of life sciences in Hamburg. And welcoming this stem cell community and encouraging us all to keep on going with our fine work. More than 3,000 people in attendance this year. And I have to say that lecture hall, that main plenary space, is really quite beautiful. No shade on Boston or LA or other years past, but those really felt like convention halls. This space felt like a opera hall or something. It must be that old world architecture. And for me, I don't know, just day one. But what struck me here in terms of thematic current at the meeting this year, it's a couple of things. One is that stem cell biology is deeper than you thought or knew. And on the flip side of that, there's a lot of technical approaches that you can use to mine those depths. At least that, I think those were the themes running through today's session, which had plenaries 1 and 2, presidential plenary, and also a couple of awards in the second. Stefan Irion, a friend of the show, he kicked it off after the second mayor. The words to kick off the meeting: the future starts here. Another thematic element here, which is, literally, technically true in any moment. It's always the future starting here. But I think, metaphorically speaking, it's that we're charging ahead into this new era of stem cell-based therapies. ISSCR current president, outgoing president, Amanda Clark, also emphasized accessibility and affordability, and shared that this is the most clinically focused meeting to date, as evidenced by the sponsorship from Bayer, but also a lot of the talks, which we're going to come back around to. The first speaker was Magdalena Götz. I think was the first speaker last year, Magdalena Zernica Götz. So they're literally going from Z to whatever Magdalena of this year's middle initial is. But she was from the University of Munich and really had some tremendous, I think, conceptual advances that she shared and that reflect the evolution of our understanding of these systems, talking specifically about organellar heterogeneity. And that's the idea that the proteins associated with cellular organelles, in other words, like, centrosomes, mitochondria, and nucleoli were the ones she highlighted in her talk, it can differ widely by cell type, also by developmental stage. She also talked about this idea that I loved, which came back, I think, in some other talks, or struck me in other talks, this idea of moonlighting proteins, and proteins that have a canonical function, but also do other work, because that's what biology does, right? It reuses the tools that have been developed and tuned over many eons of evolution. And in this case, she talked about this disease, periventricular heterotopia, and showed through all her tremendous demonstrations and data and analysis, experimental design, that the disease is not just a migratory defect, but it has its basis in a failure in differentiation. So I thought that was a nice concept that she advanced there. Also, the idea of organellar heterogeneity, I hadn't thought about in that way before, which is kind of daunting, because it's like, okay, now we got to think of everything over again, reframe every question that we've ever had. Next, and this was cool. I like the design of the presidential plenary today, because I think, it went down to these granular level things. Like in this case, Magdalena Götz talk, it was about the organelles. With Tina Mukherjee, kind of went in a different direction than we usually see, looking at, in this case, insects, starting with Drosophila and then moving in to mosquitoes. You don't see a lot of Drosophila in the stem cell studies that we focus on the show. So that I think, nor do you see it, I think, often in the meetings at the ISSCR, particularly in more recent years, as we move so much closer to the clinic. In this case, this was this idea that in Drosophila and also in mosquito, that the immune system is primed and induced by this odorant receptor pathway. And I thought this was even next level, where Dr. Mukherjee took it to clinical relevance, and talking about disease pathology. Yet don't see a lot of Drosophila or mosquito studies in the ISSCR, particularly in recent years, as we've moved closer to the clinic. Here, Dr. Mukherjee was talking about how the immune system is primed by the odorant receptor pathway in Drosophila, and then endowed that insight with clinical relevance by talking about the relationship of that olfactory immune priming with vector competency in mosquitoes. So, that the idea there, which I thought was so cool, is that these kind of these city mosquitoes, I guess you would call them, they haven't been primed in the wild by wild animals that would make them more resistant to the proliferation of the pathogens in the gut. So weirdly, ironically, it's the fact that the mosquitoes are primed only by humans that makes them so pathogenic. Then there was Catherine Plath. Again, we're talking about insects, then going a different direction here, granular down to the chromosome, and the X chromosome specifically here. And this is in the vein of like, yeah, the biology is deeper than you think. Call back to the moonlighting protein that Magdalena Götz talked about. Here, talking about exist, it doesn't just mediate inactivation of the X chromosome, that it also can bind to all autosomes, which I think has a tremendous impact. If you think about it, exists actually silencing genes on autosomal chromosomes. And not only that, but she showed that there was like sex differences in the degree to which transcription was silenced. So really important insight, and something that may have an impact on differential disease susceptibility or other aspects of sex difference in differentiation in human biology, physiology. Next, it was Sarah Teichmann. I'll note also, I love this, that our outgoing female identifying president had all female identifying speakers here, which is a nice change. I'm sure it's been done before. It won't be the last time. But I think it reflects the effort by ISSCR to really encourage a more diverse speaker lineup along gender as well as racial lines. So that's a good thing to see. Next, as I said, Sarah Teichmann, who was going with the tech element, and that's in the vein of here are the tools that you can use to mine that tremendous depth of human biology. And this is like the human cell atlas idea. And I thought using a really inspired exemplar in the human thymus, because it represents this whole kind of development and training that happens to cells within the adults. The pathway that T cells have to take to being trained during their development, and all the molecular biology and genomic, transcriptomics and whatnot that takes place over the course of that evolution. So using that as an exemplar for combining all these different data sets in these massive cell atlases that have high resolution. So yeah, I mean, for me, it was a great presidential plenary that encompassed, I think, soup to nuts at a granular level across many different model systems. And also, I think ultimately was a setup for this whole idea of how we're moving toward a better understanding of human biology, not just for our basic understanding and insight, but also with a specific mind towards therapeutic translation, which is, this meeting is jam-packed with all those. And then after that, it was on to my favorite session of the day, and what will probably be my favorite session of this entire meeting, which was, I don't know what it was called, but I have here written Stem Cells in Space. And this was a really great and well-attended session that I just have to say it. I mean, he wasn't the first to speak, but Arun dominated that session, led the group with his insights, sharing some of his results, but also, yes, some really great results and eye-opening kind of experiential discussion of what it's like to conduct experiments on the ISS. And there was a lot there. I'm going to let Arun kind of take the lead on that, but some of the things that I took away from that, which is that this is the decade of results at the ISS. So previous decade being the decade of utilization, setting it up, establishing the kind of the template for going up and doing these experiments, and we're currently in the decade of results. And what was shared at the meeting, which Arun may go into a bit more, I thought was really promising and showed a pathway toward what is the main aim of the work that's done in space, which is in part, at least, to mitigate the effects of low gravity for extended periods on human health and physiology. And for the most part, I got to be honest, it seems pretty bad. It's not good to be in space. But also there was some other kernels in there that I thought were really fascinating, mostly just about experimental design and what it takes and what it's taken. And the direction that science has taken, and a really nice primer for the live show that we're going to be doing in a couple of days, got me really excited. Arun, you did such a good job. Tell me, what do you think? How'd the session go in your view?
It's okay to say it was your favorite session. It was, I mean, you're biased, I'm biased too. It's all good. It was a lot of fun. I had to wear a different hat, not my podcast hat, but my day job hat, I suppose. It's always fun to talk about this, and it's a reflection of how, I guess, seriously the ISSCR is taking this topic, because over the last couple of years, we've had a few different uh, focus sessions on this exact topic of stem cell research in space, and in part, it's because this area is just so rapidly growing. Like what you've mentioned, the International Space Station is now, uh, it's been live for research and development for more than a decade now. The first decade was just getting it online, and then is the decade of actually, making the data happen. Now it's a decade of results, like what you're alluding to. On the panel, it's folks that I have known pretty well over the last few years, in terms of folks who are also doing the research in terms of the stem cell research on ISS, but the folks who are also enabling the projects to happen, such as Michael Roberts and Davide Morota, who's also going to be on the live show in a few days. They're from the International Space Station National Laboratory, also supported by the Center for the Advancement of Science and Space, or CASES. They're one of the NASA affiliated funding agencies that's supporting the work that's being done in space, such as the work that's done by yours truly, but also other folks on this panel were Valentina Fossati, Jean Loring, who's been on the show, couldn't be there, unfortunately. I think her flight got canceled, which is pretty brutal. I mean, that is a long flight, and for it to get canceled is really unfortunate. But we'll catch up with Jean later. Also, Kat Jameson, who's going to be on the live show in a few days with Davide. She's from down in San Diego. Uh, Honestly, we just wanted to give a flavor for the work that's being done up there. It's not just us. It's not just stem cell research. Stem cell research in space is just a small segment of the broader biomedical approach and the biomedical market, you could say, for research that's done in space. But also, there's physical sciences and material sciences research that's done on the International Space Station, and we're not even touching on that. I think, and I think the different speakers touched on these points that, mechanobiology is fundamentally altered in a microgravity context. And it makes sense. You're taking gravity out of the equation. And we know through lots of studies that have been done on Earth, stem cells are extremely sensitive to alterations in mechanobiology. And for example, stem cells are really sensitive to mechanical shear and shear stresses. They can influence their proliferation, differentiation, and so on. And so, if that mechanobiology element is altered, then yeah, of course, the proliferation rates or the differentiation rates of cells in space is going to be altered too. A lot of this is sort of context dependent. Some stem cell types, like pluripotent stem cells, respond differently to some of the cancerous cell types that Kat Jameson was talking about as well. The other element of this, I think, in addition just to the basic science that we were just glossing over, just because we wanted to give people a feel for this field as a whole. The other element of this is the tech development that's required to actually enable these projects to get off the ground and actually function in the International Space Station. These are really tough experiments to do. You have to work with a team of biomedical engineers, aerospace engineers, mechanical engineers, to custom design hardware for the most simple cell culture experiments. I know Gene and Valentina were talking about how they're able to grow multiple organoids, but they had to use a very simplified system of basically sticking the organoids into cryovials as their way to increase the scope and scale of this, and not being able to do a media exchange on these organoids for about a month, which is pretty wild. And that itself introduces a lot of experimental variability and just things to think about. But that's what you got to do in space. Unfortunately, you have to simplify and streamline the experiments that you do. So in general, just a really fun talk. It's good to catch up with other folks who are in this field. I was really pleased with the attendance, and perhaps that's because it was right after the first plenary, so everybody's fresh, which is great. But hopefully, we'll have other sessions like this focusing on research in space in the years to come as well. And then after a quick coffee break, it was on to plenary number 2, which is titled New Technologies to Engineer and Phenotype Stem Cell Systems. This was really a computationally oriented plenary, and I think it's great that they were leading off with this on the first day of ISSCR 2024. I think one of the speakers commented, I think Fabian Thies actually said how he was surprised that the computational plenary was that early, because usually they pushed on to the later portion of the meeting. I actually think that's true in a way. I forgot who said this is the most clinically oriented ISSCR. But on day 1, we haven't seen much in terms of clinical results or that sort of thing, but it's definitely coming, that's for sure. So leading off, Jonathan Weissman from MIT, he's talking about basically developing PerturbSeq, which is something that his laboratory did over a few years ago. And this is, of course, such a powerful system for genetic screening, high throughput CRISPR screening. They started with just a few genes, but ultimately turned this into a genome-wide screening methodology, which I think has been adopted by a lot of people around the world. Now, one quote that he had, which I thought was good, was, "With poor model systems, functional genomics gets us to the wrong answer faster." I think that's pretty true for anything computational. We talk about bad data sets, garbage in, garbage out. If you have a bad initial data set that's feeding in to whatever algorithm, you're going to have a bad result too. So you got to be really careful with your model systems. You got to be really careful with the data that you're actually inputting into those algorithms. And specifically, they were looking at PerturbSeq how it gives insights into the mechanism of zonation and different model systems. A really nice overview of a very, very powerful technology. Moving on to Fabian Thies from Helmholtz Munich, just from here in Germany as well. He's talking about modeling single cell dynamics across time and space. And one really cool concept was using generative AI, sort of similar to ChatGPT that we're all familiar with, right? That could be used to predict a cell's perturbational response, or a response to some sort of environmental perturbation. And he gave an example of a technology that have developed in RegVelo or reg velocity, which is enabling you to do a computational prediction of cell state, again, based on an AI predictive algorithm, right? It's able to predict knockout effects in zebrafish, neural crest. So I think that's a pretty nice application there, but you can envision how it can be used for a million different applications as well. Next, we had Suzanne Rafalski from the Allen Institute, talking about some stuff that we actually covered on the show not too long ago with Rugen and Wardane, for example. Not talking about the imaging side of it as much, but more the application of the really cool custom fluorescent reporter IPS lines that they developed at the Allen Institute. This was titled Toward a Holistic and Quantitative Stem Cell State Landscape. And it's basically looking at what do we need to know about how do we get the best understanding of cell dynamics? What are the criteria and what are the properties that we have to nail down, right? And so she mentioned there's basically four things. There's cell organization, cell function, molecular census of the interactions of the cells within the environment, and the general cell environment as well. So those four things. And like I said, they're using reporter iPSCs to really understand the dynamics of cell state as a beautiful visual readout of how things are moving around intracellularly, but also between cell populations as well. If you haven't taken a look at some of their IPS reporter lines, they're pretty amazing. It seems like every month they have a brand new fluorescent reporter IPS line tagged with a different organelle, different protein that you can use for your own research purposes. I mean, I use a number of them in my own lab. And, I always really like the Allen Institute talks because one of the core tenets of the Allen Institute, I believe, founded by Paul Allen, and I think he inspired and instilled one of these tenets, was to democratize the science that they do and try to bring it to everybody. So they talked about how they're sharing all the tools that they're developing, sharing all these IPS lines that they're making. And that's what you need. That's what you need to ultimately enable anybody to make these amazing scientific advances in our field, right? And, next up was someone who we're very familiar with here on the show and who's been, seems like coming out with a paper, a major high profile paper every single week now, using a pretty elegant model system that's relatively simple. And that's Matthias Lutolf from EPFL Lausanne. Like I said, we've had him on the show. He gave the John McNeish Memorial Lecture on engineering next generation tumoroids for precision medicine. And we've covered these papers recently on the show, and you can even listen to the interview that we had with Matthias a while back. But he was talking about how well, first he actually went back, which I really liked. He went back to the very beginning of the tumor or colon organoid field, the gut organoid field, and how Hans Clevers first described those LGR5 positive organoids and how that was, almost 2 decades ago, and how we've come so far in applying those organoids and making them, better model systems. And they're in the Lutolf lab, they're incorporating those LGR5 positive intestinal organoid cultures into the stereotyped, engineered pattern tubular organoids on chip. Seems like a lot, but if you look at the paper itself, it's a pretty simple system. It's just like a little lumen when you see the organoids on there and they're stimulated, they respond to biomechanical stress. They can have a cancer initiation phenotype on there as well. And like I said, they've had a ton of really high profile papers, really fleshing out the system, even incorporating some optogenetics into the system too. So just an amazing talk, highlighting the productivity of Matthias Lutolf and his laboratory. And finally, wrapping things up was Jun Wu, also somebody who we're very familiar with here on the show, who gave the Outstanding Young Investigator presentation on the, quote, curious cases of pluripotent stem cell adaptation.
Yeah, I mean, that I love the titles and I love the storytelling in this session. And I'll come back to June Wu's talk in just a second. But as you said, with Matthias Lutolf, going back really gave a real vision of where he's going. And yeah, I mean, he came in hot to this meeting. I think it was like 3 big papers on these mini colons in the last 3 months or so. And at the end, he dropped in. He was like, "Oh yeah," he showed like, I don't know, 8 different other microfabricated channels that combine his notion of like making and building. And it was all over the map, vasculature, airway, you name it. So I don't think he's slowing down anytime soon. We can expect a lot more from him employing these other organoid on a chip systems, because yeah, the first, right, was the intestinal organoids, and look what came of that. We talked for 4 years about organoids, and now I guess the next 4 we're going to be talking about Lutolf papers. But also in the session, yeah, going back, the Weissman talk I loved. After that PerturbSeq, I went deep, rabbit hole to see every PerturbSeq paper out there, because that's the best thing about this meeting, is that you see the papers are all out there, right? A lot of it's published, but you see the investigator convey their work, and you can see how it would apply to all these questions that spring to mind. So yeah, a lot of those questions have already been addressed, but I was thinking to my system, hey, I should PerturbSeq something. So that's on the menu. And Fabian Thies, great energy, loved his presentation and delivery. And this notion of the virtual cell, to me, was transformative. I think he was very cautious, and everybody is very cautious about how well or how realistic that is in the current state of the art. But the use case at the end with the pancreas organogenesis, where you could ask the system, what happens if I knock out this gene? How does it affect the cells? And you impute that, and it gives you an answer that matches reality. Like, that's crazy. It's coming to the point where really, legitimately, based on existing data sets out there, you can do these virtual experiments that can really guide your research. So that was a bombshell to me. And as you accurately described from the Allen Institute, the big 3 sciences, which was big science, team science, and open science. I loved that, and it reflected the tremendous ambition and how well underwritten the Allen Institute is to do all the work that they're doing and sharing. And yeah, then it was Jun Wu, who I really admire, great friend of the show, of course. But I admire him because he kept on with the thing that I gave up on in my doctoral thesis, with all his pursuit of chimerism, and he's taken it to truly next level. I mean, he had some mouse horse chimera. I mean, a mouse horse chimera, Arun, everything, rats. After the talk, he talked to my guy Dan Besser about some sheep chimera. I don't know if that's published, but he's got some sheeps in there, all kinds of non-human primates. So he's really gone deep on the chimerism, and driven by this, yeah, this idea of curiosity-based research, as he described it, as a complement to hypothesis-driven research, because that's what I really think is a common theme and inspiration behind a lot of his research, is like, what if? And I can imagine that's a really fun lab to be a part of and work to collaborate on, because it does scratch that itch where you're like, yeah, what if I put a mouse and a horse together? Let's see what would happen. Although obviously for truly, translational and basic science reasons, not just for the island of Dr. Moreau factor, truly advancing mechanistic science and our understanding of the complementarity of 2 cell types, 2 species over long time scales of evolution. So fascinating stuff. And after that, I mean, for me, I know you went to the cultured meat session. I saw you in there. So in order to give our listeners a more comprehensive view of the talks today, I went to this translation of academic research from nice to have to clinical imperative, which was, I think, nice. It was nice just to go to. Stefan, I think, gave the real, in real life, he's doing this stuff. So he had a perspective that I really valued. And that was totally realistic. I mean, there was a little of a debate element to it, where it asked questions that maybe intuitively you would think make sense. Like, hey, should an academic, I know obviously everybody who's listening to the show probably knows already the answer is no, but like, should an academic be allowed to make drugs for cheaper than an industry partner? And like, obviously no, for a bunch of legal reasons, but also just conceptually how that could like disrupt and perhaps derail the whole ecosystem. I think these are important things to consider. And more than that is, yeah, the differences in this cell-based therapy approach to the existing pharma paradigm. It's really uncertain. And when you're talking about a chronic treatment versus a curative treatment, that's a whole different ballgame, particularly when you can't really make promises in terms of like how curative that will be, which is certainly where we're at in the current state. We're still trying to establish what the efficacy of these things are. So it's at once, I think, a bit daunting, but also a testament to the tremendous progress we've made so far. And I'm thinking that maybe that talk for me was going to be the lead into a lot of talks that I have on my calendar that are really translational and clinically focused. Arun, tell me about this cultured meat talk.
Yeah, it was a really cool session. I think perhaps the first time there's been a specific session on cultured meat at the ISSCR annual meeting, in part because sort of like what's happening with the space side of things, the overall critical mass in this field is increasing, right? There's a lot of biotech companies, there's a lot of academic investigators who are getting to the bottom of how can we make, stem cell derived skeletal muscle in combination with adipose tissues to make that, that dream of cell-based meat a reality. Like, how do you improve the differentiation, the cell culture costs, reduce the cell culture costs, make it scalable? How do you do all these things across not just cows and pigs, but also potentially fish? Like, how do you address some of these concerns? I mean, this is something that was really exciting to me, and I felt like it's one of those areas in stem cell biology which is almost immediately translatable, right? And not every field in stem cell biology is that way, but sometimes, and you got a sense of that in this session. Some of the scientists, such as like Paul Burridge, who I know really well through my time in Joe Wu's lab, his whole thing has been making media, cell culture media, cheap and easy to make, right? And he was talking about a situation where he published a paper on negligible cost chemically defined media, and the only people in the biotech industry who were attracted to that paper were people in the cell-based meat industry. Okay, and so he immediately turned a light bulb on in his head, and he was like, okay, maybe this is what I should be applying this technology for, applying this paper for. So it's immediately translatable field in a lot of ways, I think. And I wanted to get a broad overview of what people are working on in this field. And it's not just one portion of the world that has, cell-based meat locked down. We had great representation of folks from the United States, like Paul, but also folks from Israel, Singapore, Japan. There are countries all over the world working in this area. So, for example, we had Shulamit Levenberg, who's the founder of Aleph Farms, which is a biotech coming from the Technion in Israel. She was talking about novel cell sources for meat production via tissue engineering, and gave a really cool story about how her lab developed this really nice protein scaffold, a soy-based protein scaffold for 3D bovine skeletal muscle, which is, an academic paper. And the application was pretty immediate because if you can make soy-based protein scaffolds for 3D bovine skeletal muscle, the next thing is you're going to try to make cultivated meat from that. So they founded Aleph Farms based on that tech. The first product was this really nice thin cut cell based steak. They addressed some of the scalability of bovine pluripotent stem cells. And it was actually the first cultured beef product in the world. I did not realize that to get regulatory approval. She also talked about the bioprinting of the scaffold and the cells and this concept of an edible bioink. Everything that you have to do in this field has to be for quote GMP, right? These products are going to be edible. They're going to be going to people. You have to make sure things are safe, things are tasty too, right? That's an important part of it. Also talking about the fat element of it because it's not just the muscle. You got to have the right ratio of the adipocytes to the muscle to make these things actually taste good. I haven't tried cultivated meat yet. I kind of do, just out of curiosity. But they're saying, and pretty much all the talks were saying, they did blind taste tests, and whether it was the pig, whether it was the pork, the tastes were pretty similar according to professional tasters. I would love to be a professional taster one day. Moving on to Shigeki Suji from A-Star, that Singapore initiative with an amazing biomedical research presence. This is the Singapore Institute for Food. He was talking about cultivated fish fat as a novel food source. Like I was talking about, you have to have that right ratio of fat to skeletal muscle or fat to meat, right? And he's talking about how you could use fish fat, which might be advantageous because of omega-3s and all that kind of stuff, right? But they derived fish adipose cell lines from a bunch of different fish types. This is really cool, from catfish, sea bass, perch, and did RNA sequencing analysis comparing the real thing with the stem cell derived cells. And they did the tasting, of course, but also testing for heavy metals, allergens, and everything was comparable or less than the real thing, which is great, except for the taste. The taste was the same. And then they incorporated the 3D culture aspect of it. Christine Troutman from ETA Zurich talking about optimized bovine myogenesis in vitro. She was a PhD student who has transitioned into the biotech industry, talking about her thesis project, which is optimizing skeletal muscle isolation, going from the bioreactors ultimately to steak production down the road. And it's a really important topic. Like, how do you get these myoblasts, these skeletal myoblasts, to mature into the myotubes that ultimately, like I'm saying again and again, taste good and have the right texture? So you have to improve that differentiation in the appropriate way. Next up was Maria Fernandez from Meatable, talking about the harvesting of potential stem cells for the cultivated meat industry. Same idea, making myoblasts better, making adipocytes better, making the combination of the two the best. And she was focusing more on the porcine, the pig side of it, porcine pluripotent stem cells. She had this really cool accelerated muscle differentiation process in the pig, where apparently she can make porcine myoblasts within 4 days, less than a week, using a doxycycline inducible system, which maybe is not as easy to bring into the market because it's a dox inducible system, but it's super fast. And we know scalability is a concern. We know by accelerating these timelines, you can make things cheaper and easier, right? And of course, finally there was a panel session with Paul Burridge, but also Nirai Govindaswamy, Anissa Chand, and Michelle Hauser. I believe there was some representation from Nature Foods, which is one of the biggest journals in this field. So it was a really nice session talking about the pros and cons of this industry, what it's going to take for this cultivated meat industry to surpass the existing industry of meat that's been around obviously for thousands of years. And in my mind, it's not just a one-to-one. You don't have to, I think these cultivated meat products have to be way cheaper than the alternative to really take over the market. But there's a lot of very interesting subtopics in this area. There's the ethical side of it. There's the sustainability side of it. There's the economic side of it. So it's just a really fun session that I haven't seen before at ISSCR.
Wow, man. I'm jealous because that was a juicy session, and I did mean that terrible pun. That's my terrible pun of the episode. But wow, that sounds like a plenary, and they really how many speakers do they have? Amazing, all that information, and the thing with it is that I've talked with your boy Paul a bit about this, and he's convinced me. To be honest, he understands the problem so well. But there's so many haters about cultured meat, so I'm a bit jealous that I didn't get to see that positive side of it because they've really thought about the problems, right? I always picture just the contamination. I mean, that's the
Yeah.
thing, no immune system, but yeah, they thought of that too. Amazing session that I missed, unfortunately. And as I'm looking at it, I'm sorry to say, the first concurrent tomorrow, actually 8:30. I'm sorry, not sorry, joking about being sorry, but I talked about how it's going to kick off to the clinical translational slide. Plenary 3 is exploring the basic principles of development using STEMCELL.
There you go.
So we're staying basic, but I'm psyched for that. We're all developmental biologists. And then we got plenary 4, which is going to be heavy clinic, tissue architecture and stem cell function in health and disease, Arun. So that's probably the bridge. It's going to be all clinical after that. But honestly, I don't care. I've loved today. I loved what they set up, and I think a really great start to the meeting. And moving on, we will have day 2 with 2 plenaries and a concurrent and a couple other sessions peppered in there. But that's it for day 1. That brings us to the end of our first ISSCR 2024 episode. Don't forget to follow us on X at STEMCELL Podcast to find out what we're up to at the meeting. A lot on, so I'm posting all over social. Watch out, guys. And visit us at the STEMCELL Podcast booth in the exhibit hall to participate in some recordings in our on-site studio, which we had at the meeting. And we love that part of every meeting we've been doing for the last 2 years, where we get to talk to the actual attendees, mostly trainees, the best part about that. Also check back here tomorrow for another episode recapping day 2 of the meeting. Until then, thank you guys so much for listening.